Welcome, everybody. I'm thrilled to walk you through the journey of building a biomedical knowledge graph today. It's pretty funny, but last year was there were two major trends on my there were two major trends on excuse me for a moment. Okay, so I'm sorry. I had a few issues. So yeah, last year was basically my LinkedIn who had two major trends. These were knowledge graphs and COVID-19. And actually today, we are just going to match these trends and we are going to cover both. So maybe a few informations about me. So my name is Konrad. And actually, as you can see in here, I have I have quite a broad experience, but most of it I was working in pharmaceutical companies. So, and maybe you can't see it in here, but actually I had a working experience with knowledge graphs. So when I was working as a mobile app developer, actually I had the chance to be working in a project which involves working with knowledge graphs, where actually we were just building an enterprise knowledge graph. Currently, I'm a data science software engineer at Roche. And so maybe a little bit more information about Roche. So I'm actually in RGITSC department, which is Roche Global IT Solutions Center. And so this is a division of Roche that supports, it is IT division, which supports most of the Roche activities from IT perspective. I have to say that this project has been built completely outside of Roche and is not part of any Roche projects or activities. But it doesn't mean that we don't do knowledge graphs at Roche. And actually today there is a pretty interesting presentation given by David, who will be talking about his use cases of using knowledge graphs at Roche. So I really encourage you to join that. And if you want to know a little bit more about Roche, I really encourage you to visit our LinkedIn website, to visit our Facebook page, or visit our main website, it.roche.pl. So what we are going to talk about today, so we'll just briefly talk about BioGrakn COVID, what it is, and what's the team that has been working on it. Then I will move to data sources and ontologies, so what data we brought to our knowledge graph. I will briefly talk about knowledge extraction, and so how you can extract knowledge, especially from unstructured data sources, and what challenges we faced during building BioGrakn COVID. And at the end, I will talk about analytical reasoning of a knowledge graph, so how you can analyze and do some analysis on knowledge graph. So BioGrakn COVID, this is a project, it is an open source project, which I've been working on with Tomas Sabat from Grakn. And this project is a knowledge graph centered around COVID-19. So quickly about the graph representation and notation. On the left-hand side, you can see a normal graph. I will use this representation whenever I talk about general graph. And on the right-hand side, you can see a hypergraph representation, which is used by Grakn. So, hypergraph, this is the extension of a graph or generalization of a graph, where one edge can link several nodes. And in here, we have these rectangular shapes. These are nodes or entities, and these diamonds represents relations or hyperrelations. Okay, so let's jump into data sources and ontologies that we used. As you can see in here, there are plenty of them, and these are pretty big. So one of the biggest one we have UniProt, where we've got proteins and genes from. We have clinical trials. We have Reactome. We have PubMed, quite a huge data source with plenty of medical publications and many others. In here, I just listed or enumerated the data sources and mappings that we have. I'm not going to go through all of them. You can just check these mappings later on. If you are interested, you can also find it in our repo. This is the next, this is the continuation of these data sources and mappings. But first, before we go into actually ingressing our data to our knowledge graph, let's quickly talk about data quality, as I can tell you that you really want to keep your data quality in your knowledge graph. You don't want to have any data discrepancies. You don't want to have several nodes representing the same entities, as this will affect and bias your analysis in the future. So how we can approach the data quality, how we can sort that issue. There is a data source called UMLS, which is Unified Medical Language System, and this is a really huge data source. And today we will focus only on two subsets of it. So one of them is Metathesaurus, which contains the biomedical entities on different levels and of different types, like proteins, genes, diseases, drugs, and also relationships between them. So as you can see in here, these are sample data. They doesn't have to be real, but so that you can feel or get the sense of it. So on the right-hand side, you can see that we have protein 1, protein 2, and there is the interact with relationship name between them. This Metathesaurus also contains a concept type, so that we have the name of a specific concept like ACE, but it will also provide you the name of the type like gene. There is also another subset of the data, semantic network, which is rather focused around taxonomy, so that you have each concept is some kind of, you have a taxonomy for each of the concepts. So like in here, on the right-hand side, you can see the graph visualization. So you can have a protein, which is of type, let's say it is a compound. So you can get this taxonomy from semantic network. And believe me, you can see it later on. You will see it later on, but it's pretty useful. Okay, so we covered data quality, where we can get it from. So let's move on to structured data, ingesting them. But first, we have to create our schema, our domain model. So in here, right now, we are creating biomedical domain, biomedical model. So how we did it? So we just identified several entities. So we have proteins, transcripts, genes, pathways, viruses, tissues, drugs, and diseases, and the relationships between them that we can actually derive from our data sources. Once we've done that, we can go to our data sources, and in this example, we will have a look at the UniProt data. And so in here, you can see that we have data from UniProt, which also contains some data from Ensemble. So we have to just identify the relevant columns. Then we have to have a look at our schema and identify the relevant entities that we would like to populate the data for. Map the relevant columns to the relevant nodes from the schema. Then once we've done that, we can just populate the attributes for each of the entity. So in here, I will just use, whenever I say entity, I will use entity interchangeably with nodes. So right now, after this process, we just populated the data for biomedical schema that we have. But the problem is that these data for this specific data sources are not updated very often. Some of them are updated on a yearly basis. And so this is the time actually when we can move to another data source or stage or part of our schema. So first, we have to define a schema, right? I was talking about papers. This is what we did in BioGrakn COVID. So I created a subdomain model, let's say, papers. And here it is. We have journals, which are related to papers, which are related to authors, to authors, which are related to organizations. Now we have to somehow link our models. We want to be able to traverse through the whole graph. So this is how we approached it. This is quite real-world relationship. These are real-world relationships. So we have biomedical entities related to paper, where the relationship type is mentioning, basically mentioning. Whenever there is something is mentioned, being mentioned, we just create a relationship unrelated to a given paper. What are the challenges that you can struggle with? So first, extracting biomedical entities from text, right? So you can identify some publicly available publications, but you have to somehow derive this text. Another problem is that, okay, you derive text, you have to somehow connect it to your ontology, right? We have UniProt. UniProts has some specific identifiers. The same is true for I don't know, for UMLS and any other ontology. So how we can approach it? First, there is no point of reinventing the wheel. So the best option is just to use already existing corpora. So what we did, we used CodeNER and SemMed. So just for those of you who are not familiar with NER, it stands for Named Entity Recognition, and this is the NLP, natural language processing algorithm, which can identify named entities, like the actual words, biomedical, let's say words like specific genes, out of text. Another way is just to extract biomedical entities directly from text, because sometimes you can find out that not all of the textual data are already mapped to named entities. So starting from the CodeNER, this is a data source which was created by people in UI/UC, which contains already derived named entities. So they created an algorithm. They ran for NER. They ran this algorithm on publicly available publications, and the output is just these entities. But furthermore, these entities are mapped against UMLS. So you can see that it will help us to keep consistency and data quality in our knowledge graph. So we can derive out of it. So we have these named entities and their types. Like in here, we have ACE2 and we have gene or genome. So it tells us what type of entity it is. Then we can map it to our knowledge graph, to our schema. So this is what it looks like. The actual data are stored in JSON files. And what we have to do, we have to just identify the relevant entities or the relevant fields that we have in our schema, that we defined in our schema. And we have to just map them. So in here, we have authors, journals, we have titles, and we have entities. So this is all about CodeNER. Now let's move to SemMed. I think that this is one of the best data sources in biomedical domain, and I talked with several people from life sciences, and I saw that not everyone is actually aware of it. So SemMed, it contains the actual entities derived from PubMed publications available in PubMed. And these entities are stored in semantic triples. So the problem that we had with CodeNER is that CodeNER has these named entities derived, but it doesn't have linkages between them. SemMed does. So in this case, we have these semantic triples, we have subject, predicate, object, and furthermore, all of them are mapped against Metathesaurus in UMLS, which once again helps us to keep consistent naming convention in our knowledge graph. So what the data looks like, I just did in here, I just did a join of two tables, and you can see that we have data for subject, predicate, and object. In here, we have NDUFS 8, which is, you can see GNGM. This stands for gene or genome. This is a type defined in or derived from UMLS, which is very helpful. You can see object, which is NDUFS 7, which is also gene or genome. And we have a predicate, interact with. So you can see that we have we can derive the linkages naturally from the text for these entities. So when it comes to mapping these entities to our model, So we can basically take the genes or any biomedical entity, map it to our schema, to nodes in our schema, to our entities, and we can take a predicate and map it to a specific relationship or edge. So, for example, for genes, there are, I think, something about 18 relationships, different predicate types. So we just defined 18 relationships between genes and then map them accordingly. So as you can see in here, NUFS8, it will be actually a gene, and UFS7 will be another gene, and interact with, so we identify this relationship between them, will be an interaction. So not all of the data, or not all of the corpora are available in online. I mean, not for all of the corpora we have the NER output. So sometimes we have to just derive data out of text on our own. So the challenge that we had was actually during working with clinical trials data. So clinical trials data, I downloaded the format of clinical trials data is XML. And so there are interesting fields in it. One of them is drug field. But unfortunately, drug field doesn't necessarily has to contain the one specific canonical name for a drug. Sometimes it contains two drug or compounds, or chemicals. Sometimes it can even contains some additional information. So how we approached it, we used SciSpacey. SciSpacey is a library in Python, and it is built on spacing, and it uses a transformers model. It has a transformer model trained on publicly available publications, and with a task NER. So we can use it to identify named entities. And as you can see in this example, it identified two entities. Furthermore, even better, SciSpacey maps data against UMLS. Once again, UMLS, but this is great because, as previously, all of our data has consistent naming convention. Furthermore, it provides us, from UMLS, it fetches this TY, which is type. And as far as I remember, I can't remember, but I think that in this case, T121 is a pharmaceutical substance, I think. So we can filter through TYs to get the relevant or the entities that we want. We can also get a score, so how sure you are that the identified entities is actually what this text represents. Okay, sometimes another problem. Sometimes you would like to get an ID from some ontology that is not available in some subset of data that we are working on. So then, if we have a proper name, like some kind of canonical name, let's call it, for an entity, we can just reach out. There are several APIs, and for example, there is RxNorm API. RxNorm is a data source or data set for a database for drugs, but they also serve API, and we can reach to one of the endpoint, which will, with the name, it will give us back RxNorm ID, which we can use later on to reach out to another endpoint and get back the ID that we want. So in this case, it is for example, drug bank ID, but it contains SNOMED, it contains mesh, and many others. So the output looks like this. For example, we have the clinical trial. We have identified canonical names for identified entities. So here, drugs. We have types, so we can filter it as we want. And we have CY. CY is a unique concept identifier in UMLS. We have score, and we have the ontology ID or ID from another ontology that we want. So let's say in this way, we just ingested all of the data that we wanted. So how we can actually traverse the graph or get some information from the graph. So in here, as you can see, reading from the left-hand side, we are looking for a virus, let's say SARS, and we want to find a gene which is related to this virus, which is the host gene for this virus. Then we want to find another gene which stimulates previously found gene, and also we want to find a drug which interacts with this second gene. On top of that, we want to find out a publication where the relationship between genes was mentioned. And you can see that in that case, you can just easily traverse the graph, limiting the exploration space, or space for exploration for possible, I don't know, drugs, a target for drugs, for possible information, just finding information about specific virus, going through the chains of just traversing the graph. Another use case, if you want to know more about clinical trials, like clinical trials, we can query it from our graph too. So we can look for a specific clinical trial. We can find the organizations that take part in this trial, or also we can find identify sponsoring organization. We can find targeted drug. We can also, and furthermore, we can link these information to all of the information about drugs and drug genes relationships, drug diseases relationships, and just traverse it and explore the possibility, the possible targets for this clinical study. Okay, so this is about just creating biomedical knowledge graph and traversing it. But let's move on to quite interesting subject, which is analytics on graph and how we can perform it. But before I start, let me just provide you a little bit of background. So it is proven in researches, it is proven in practice in many domains that nodes tend to cluster or tend to create communities with other nodes that share similar functions in a specific network or the whole system. And so based on this, we can derive, we can quickly come up that function of a specific node in a network can be derived from this node's n-hop egonet or neighborhood. And this is true, for example, for protein-protein interaction network. So PPI, protein-protein interaction network, is composed of proteins as nodes, and edges in this example are the interactions between proteins. And it just, it's well established that proteins, in this case, nodes that represents proteins tends to cluster or create communities with other nodes that has similar functions, like in here, metabolic processes or inflammation and immunity. Okay, so we have, about tasks and task types. So we have three major task types. We can train a model for node classification, for link prediction, and graph classification. Today I will focus mostly on link prediction, as this is, I think, on this graph it is the most exciting task. So what is link prediction and how it works? So in our graph we have nodes. In knowledge graph they can have different types, and we have some existing edges between them. But the assumption is, this is a true assumption, that our graph is notoriously incomplete. And we would like to find some possible edges that we are missing. So we have a considered edge, we consider some edge, and we would like to check if this edge actually should exist in our graph. And so how you can approach it? One of the methods, pretty cool methods and quite popular recently, are graph neural networks. And I would like to show you some very initial results of the analysis done with graph neural networks on BioGrakn COVID. But first, before we move in any further, let's go back to link prediction. In neural networks and in machine learning, many times we have a term negative sampling. And it is it means that we basically draw a random sample from our data, from our data set that is negative, that doesn't exist, that it's not true. So in this case, we are just randomly selecting two nodes between which there is no relationship in our graph. Obviously, you can notice that it is a bit biased, because previously I said that our graph is notoriously incomplete. So maybe the edge that we selected is actually a true edge, which should exist. But in fact, in practice, this solution works pretty well. So how I approach it. So as an input, I took structural features and eigenvectors of adjacency matrix, which represents some latent, hidden structural features of a graph of nodes. And as models, I took plain vanilla graph convolutional neural networks with positive negative sample split ratio 50-50. So the output, maybe at first, graph neural nets as the output gives us probabilities, as the last layer of the neural net is softmax function. So we've got the probabilities, which which tells us if an edge is true or not. So I selected the threshold for probabilities to say that a given edge is true to be equal or to give me recall of 85%. And in this chart, you can see that this chart represents a precision recall versus threshold, and this 85% recall or threshold is actually marked with the red dot. Let's move to confusion matrix, and it provides us a little bit more information. So recall 85%. We've got a precision of 91% and specificity more than 90% with this model. And actually, this is what I received, what I got on my test dataset. So a dataset that wasn't seen by a model during the training. So this is pretty cool. Looks pretty good, but you have to remember that as negative edges, we used random edges, which sometimes might be edges that are not valid in our schema. So let's say like drug transcription, or even going even further, it's like getting drug owns a company, let's say. But still, it is not. It's in practice, it works pretty well. But when I randomly sampled edges that doesn't exist in our graph for genes, it turned out that 76% of them are marked as positive, which is not probably it's not the best outcome. But when I got top 5 predictions of these, it turned out that 3 genes relations co-appeared. I mean, 3 genes for these edges co-appeared in papers targeting the same entity, I know, compound or other gene. So it tells us something. So then I added to a compute graph, not to the GNN yet. I have to get more data, but I added to a compute graph drug disease interactions, and then I computed the same, and I've got a prediction of 22. So I've got the negative edges that doesn't exist in our graph, and it provides me 22% of it predicted 22% of edges as positive. Which actually, when I got top 5 predictions, it turned out that 2 of these drug disease interactions actually exist and can be found in biomedical papers, and these drug diseases exist. So it shows you how you can complete your graph, how you can work, how you can analyze your graph and complete your graph, find the information to complete your graph. So about future work, I'd like to probably generate more structural features, add some word embeddings as a features, probably change the negative sampling distribution, try different models like shallow embeddings and graph convolutional nets, and probably find out models to run hypergraphs natively, models on hypergraphs natively. In terms of data model, I'd like to add drug-drug interactions, add possibly some patent data, and UMLS ontology. So you can see, I just showed you how you can build a knowledge graph, how you can traverse and get some information out of it, and how you can complete knowledge graph or do some analysis on it. And such knowledge graph can be used by, I know, scientists to find out maybe new solutions, some new drugs or new targets to cure, let's say, COVID-19. In here, in this screen, you can see the summary. So graphic numbers. So we have more than 300,000 of nodes and more than 2 million edges. I know that there was quite a bit of information today. I hope that you enjoyed it, that you learned something, and I would like to encourage you to visit the specific channel for Q&A on Cadence, and I will try to answer all of these questions there. Thank you for joining today's speech.